Hi everyone, and welcome to Regents Review Session 5. This is Ms. Winans. The topic we'll cover today is reproduction. Cancer is considered mitosis out of control. So your cells are really efficient at making sure that they go through cell division correctly. There are several checkpoints throughout the process in which the cell can actually realize that there's something went wrong, there was a mistake made, and it can trigger what is called apoptosis. Apoptosis is what is called programmed cell death. If you remember back from cell biology, we learned about lysosomes. Lysosomes are organelles inside the cell that contain enzymes. What will happen during apoptosis would be that the cell would recognize that there's something really wrong with it and it's not supposed to reproduce. So what will happen is it will receive a signal that will tell the lysosomes to pop. All of those enzymes will be released and they will start eating the cell from the inside out. So literally the cell will implode and disappear, which is a good thing because there's something wrong with it. So for the most part, every day our cells that are dividing are going to go through a checks and balances system. And that check system is going to say this is a good cell, let it remain. This is a bad cell, it has to go. Unfortunately, the signal that tells the cell that it's supposed to go through apoptosis is coded in the DNA. And as we know, everything in the DNA is vulnerable to a random mutation. And that's what cancer is. The signaling cascade that signals apoptosis is mutated in cancer. And unfortunately, instead of telling that cell there's something wrong with you, it's time for you to go, it says there's something wrong with you, it's time to reproduce. And what happens is a cell that only knows how to reproduce and doesn't know how to maintain homeostasis starts reproducing and reproducing and reproducing, and it forms a tumor. These tumor cells can then break off into the bloodstream and spread, which is called metastasis. Once the cancer metastasizes, it gets more and more dangerous. The biggest problem with cancer cells is, again, they only know how to reproduce. They don't know how to maintain homeostasis. So what happens is these cells, it looks like these are skin cells. These skin cells over here, which know how to maintain homeostasis, can't compete with the fast reproduction and the ability to control the nutrients of the cancer cells. So eventually what happens, if there's more cancer cells than there are healthy cells, then the function of that organ stops, and then the person starts not to feel well, they can't maintain homeostasis, and unfortunately that's when we start to detect it. The main treatment is to remove the tumor. So they'll cut this out, remove the tumor, and then what they'll do is they'll either treat the person with radiation, which is very direct. It goes right to the location and tries to kill the cancer cells in that area. For cancers that tend to metastasize quickly, spread quickly, they use chemotherapy. Chemotherapy is pretty aggressive. It's chemicals, and the chemicals don't really know how to distinguish the difference between healthy cells and cancer cells. So while the chemotherapy is doing its best at trying to kill all the cancer cells, it can kill some healthy cells in its wake, and that's why people tend to lose their hair when they're on chemotherapy. But again, cancer is mitosis out of control. It's when a cell it divides, there's a mutation in it, and it's not supposed to continue to divide. It was supposed to go through apoptosis, but it didn't. Hope this helps. In the modeling mitosis lab, you used pipe cleaners to model the stages of mitosis. Remember, the stages of mitosis are PMAT. Prophase, appear, metaphase, middle, anaphase, apart, telophase, 2 new nuclei. We start in interphase. This is the phase in which the cell spends most of its life. So I'm going to draw some chromosomes. Okay, so we're going to notice that this cell has 1, 2, 3, 4, 5, 6 chromosomes. Okay, so if we're about to divide into 2 cells, the first thing we have to do is replicate all of our DNA. And in previous slides, you learned that that happened during S phase. So when we go into prophase, this is the first time we're going to see that X shape of the chromosome. With 2 sister chromatids. Remember, chromatids are identical copies of genetic information. Chromatids are imperative to mitosis because remember, if we're going from 1 cell to 2 cells, we want to make sure that both cells have a full set of DNA. So instead of seeing the single line shape of a chromosome, we now see the X shape of the chromosome, and that's because we successfully replicated the DNA during S phase of interphase. So what happens in prophase is that the chromosomes appear. In metaphase, the chromosomes line up in a single file in the middle of the cell. In anaphase, we pull those sister chromatids apart because remember, those sister chromatids are just, identical copies of DNA. So this is just a way that we ensure that both cells get a full set of DNA. Okay, and then we go to telophase, in which the cells now have, I didn't finish that cell. Have the chromatids separated to opposite sides of the cell. Okay, and then if it's an animal cell, the cell will go through cytokinesis and split into 2 cells. If it's a plant cell, it'll just build the cell plate, which eventually will be the cell wall. But if we notice, now that we take a look at these cells, they're identical to the original cell. Both cells have 6 chromosomes, which means in mitosis, we successfully made 2 identical body cells. Okay. All right, so now what we're going to do is we're going to demonstrate meiosis, which is a little different from mitosis. Meiosis makes sex cells. Remember, sex cells will only contain half the genetic information. So at some point, we have to cut the genetic information in half. And that's because the goal of gamete, which is either a sperm or an egg, is to unite with another gamete to create a body cell. The first body cell of life is called a zygote. So we're going to start with the same cell that we had in mitosis. Okay, and remember, in interphase we go through S phase. That's when we replicate the DNA. So when we enter prophase, we will now see those sister chromatids. So we'll see that X shape of the chromosome. Okay, so this is going to be prophase. Now, remember PMAT? Okay, so we have prophase, appear, metaphase, middle, anaphase, apart, telophase, 2 new nuclei. This is going to happen twice in meiosis, and I'm going to skip a couple phases in my drawing, so I don't want you to get confused. The most important step in meiosis, and this is what makes meiosis different from mitosis, is metaphase, okay? In metaphase 1, the chromosomes are going to line up in their homologous pairs. Homo means same. Homologous chromosomes are chromosomes that contain the same genetic information. They were originally inherited by the original parent's mother and father. We're representing homologous chromosomes using color in this particular demonstration. Other times they'll represent homologous chromosomes using size. So in prophase, the chromosomes appear. In metaphase, the chromosomes line up in the middle. And notice how what's happening here is that the chromosomes are in the middle, but they're in a double file of homologous pairs. So the first round of meiosis, we're going to separate the homologous pairs. This is going to allow us to go to cut the chromosome number in half. So what's going to happen is instead of pulling sister chromatids, we're going to pull homologous chromosomes to opposite sides. So when we're in anaphase, we're going to start moving the homologous chromosomes apart, because remember, we want to cut the chromosome number in half. We go into telophase, and that allows us to successfully cut the chromosome number in half. But we still went through S phase. We still have sister chromatids. So since we still have sister chromatids, we need to also pull those chromosomes apart. So then we go through metaphase again, anaphase again, and by the time we get to telophase 2, we separate the chromatids. So again, let's line these guys up in the middle so we can see what metaphase 2 looks like. Okay, so again, if this was metaphase 1, notice I left out prophase. Prophase still occurs. I just left it out in my drawing. So this is metaphase 1. This is metaphase 2. This is anaphase 1. Okay, so in anaphase 2, we're going to separate those chromatids, because remember, the chromatids are still copies, so we want to separate them because we only want one copy of each chromosome. And so we actually, at the end of meiosis, end up with 4 cells, which is different from mitosis. And what we end up with are four cells that have half of the original genetic information. apart, because remember, we want to cut the chromosome number in half. We go into telophase, and that allows us to successfully cut the chromosome number in half. But we still went through S phase. We still have sister chromatids. So since we still have sister chromatids, we need to also pull those chromosomes apart. So then we go through metaphase again, anaphase again, and by the time we get to telophase 2, we separate the chromatids. So again, let's line these guys up in the middle so we can see what metaphase 2 looks like. Okay, so again, if this was metaphase 1, notice I left out prophase. Prophase still occurs. I just left it out in my drawing. So this is metaphase 1. This is metaphase 2. This is anaphase 1. Okay, so in anaphase 2, we're going to separate those chromatids, because remember, the chromatids are still copies, so we want to separate them because we only want one copy of each chromosome. And so we actually, at the end of meiosis, end up with 4 cells, which is different from mitosis. And what we end up with are 4 cells that have half of the original genetic information.